Hello again. In this penultimate video, we are going to create new life. So, the fertilization process, the sperm needs to meet the secondary oocyte. And here's an interesting tidbit. The fastest sperm, Mr. Michael Phelps, the fastest swimmer, is not necessarily the baby daddy. Okay, so the first thing that needs to happen is the sperm needs to penetrate through the corona radiata, because remember the secondary oocyte has the zona pellucida around it, and then it had follicular cells forming the corona radiata to protect it while it was tumbling around and made its way into the distal part of the fallopian tube. Fertilization normally occurs in the distal third of the fallopian tube. After the sperm has penetrated the corona radiata, do you remember the acrosome on the tip of the head of the sperm? Well, that acrosome is going to release its digestive enzymes, and that's going to cause a momentary change in the electrical balance of that zona pellucida. And in that momentary change, a sperm is going to be able to get through that elastic acellular substance. It's probably not going to be the sperm that released the acrosome, the digestive enzyme. That sperm probably going to get digested with the enzymes. So it could be a nearby sperm, it could be a sperm clear on the other side of the oocyte, but some sperm is going to get through the zona pellucida, getting it penetrated through it. So now we see a sperm on the inside, and what needs to happen next is the plasma membrane surrounding that sperm is going to fuse with the plasma membrane surrounding that secondary oocyte. Once those two have fused together, then the nucleus inside the head of the sperm will enter inside the cytoplasm of the oocyte. The rest of the sperm not needed. Not going to contribute a thing. Okay. So now that the oocyte has officially been fertilized, that triggers meiosis 2 to finish. So remember, it was stopped in metaphase 2. So it'll have anaphase, it'll have telophase with cytokinesis. So we will have a second polar body formed. All right. So we had an oocyte. The sperm came in. At the end, we are going to end up with two polar bodies. All right. So here is what we would look like. We have two polar bodies at the end of meiosis 2, and we would have two separate nuclei still. Then the two nuclei, and each nucleus is N, they are still haploid. Then the two nuclei need to fuse together to make a diploid cell. And that would form the zygote. Okay, so when it forms the zygote, we still can have sperm on the outside. We still gonna have zona pellucida and corona radiata on the outside. So the two nuclei will fuse together. We're all done with all of that, and we will end up with one cell with one nucleus. Okay, from the father, only the nucleus becomes part of the zygote. The father only is going to contribute DNA. The mother is contributing cytoplasm and all the organelles. Everything is coming from the mother. That's why the mom cell is so big, because she's contributing everything. 2 cells worth of everything. And that is why mitochondrial DNA can be used to determine maternal lines, because the mitochondria are only coming from the mom. All right. So this is how 1 + 1 makes 3. We start with a secondary oocyte from the mom, can live a max of 24 hours. A lot of them only live 6 to 12 hours. We end up with a fertilized oocyte where we can see 2 polar bodies, usually. Sometimes we have 3. We can see 2 nuclei. They're called pronuclei because technically, yes, it's a nucleus, from the oocyte and a nucleus from the sperm. But, now we're making a zygote, so not going to be that yet. And this one, you clearly can see the zona pellucida on the outside. And then over here in the left lower quadrant, this is a 2-cell embryo. Technically, it's not an embryo. Technically, it's a zygote. I wrote the wrong word here. It's a 2-cell zygote. Where you can see zona pellucida still present, and you can see some corona radiata still present. All right. Now, if we have infertility problems, we can have in vitro fertilization. And this is an example of in vitro fertilization where they are taking the nucleus of the sperm and jabbing it inside the oocyte of the female. And you can clearly see there's a polar body from before, and there is a zona pellucida present outside the oocyte. So, fertilization in the distal third of the fallopian tube. This is before fertilization. Fertilization's occurring here. You can see all the little sperm afterwards. Then we have pre-embryonic development. Like I said, I used that word embryo incorrectly. We have a zygote. We go from 1 cell to 2 cells, and then we end up having a ball of cells, multicellular zygote traveling down the fallopian tube towards the uterus. Takes multiple days going down there. Notice there's no flagellum. It's relying on the cilia to create currents, a little bit of peristalsis to move it down towards the uterus. During this timeline is when this can split and form identical twins. Because remember, fraternal twins are with 2 sperms fertilizing 2 oocytes. This would be identical twins with this 1 ball of cells would split into 2. Okay. Starting week 2. So around the time of implantation, that's when we start calling it an embryo. So an embryo is going to be implanted into that very fluffy pillow top mattress of endometrium. And this can cause spotting. A woman can confuse it for a light period at the time. And the embryonic development, we call it an embryo all the way from the beginning of week 2 through the end of the 2nd month. And so here at the end of 2 months, 8 weeks, this is what an embryo looks like. And you can generally recognize this as being human. For like 6 weeks, you can't, because what a chicken looks like, what a frog looks like, they all look like the same thing as what human looks like. But by 8 weeks, you can generally recognize this as human, although there's still a lot that needs to be developed. All the organ systems have started being established, but they're not functioning. Okay. So even though you may say the heart is beating, the heart is technically beating, but it is not circulating blood around. The cardiovascular system is not functioning like our cardiovascular system is functioning. Okay. Then the beginning of the 3rd month through the rest of gestation, it is called a fetus. At the end of the 1st trimester, that's when the placenta starts taking over. So we have a fetus with a placenta going to start taking over hormone production. So that corpus luteum gets quite huge because that is what's maintaining the pregnancy for 12 weeks. Okay. And now the organs are continuing to develop and will start functioning. A lot of them will not be fully functioning. Like, for instance, the lungs are not fully functioning until around 36 to 38 weeks gestation. Okay. The placenta is not fully formed until 20 weeks. Okay. So if you have placental failure at 4 months, you absolutely can have a late spontaneous miscarriage, what's called a spontaneous abortion. So the last half of pregnancy, basically the fetus is increasing in size and gaining a lot of weight. The last month, the fetus is gaining a half a pound a week. It's quite significant. And movement will start occurring in the last half of pregnancy as well. As far as when labor occurs, well, it's the fetal hormones that are going to be triggering labor, not mom's hormones. Okay. So if we look at the pregnancy, remember development starts while still in the fallopian tube. We start out with a zygote. It's going to start kicking out that hormone HCG, which is going to keep the corpus luteum producing lots of progesterone and estrogen. At 13 days, so this is after implantation, this is what the embryo looks like. So you see how it doesn't look anything like a human. At 5 weeks, this is what it looks like. Not really that human. We're just getting a little limb budding happening. 9 weeks, much more human looking. And then this is up to 20 weeks gestation. At 21 weeks, there are a few procedures that can be done actually on a fetus while pregnant still. There really aren't any pictures of babies late in pregnancy because there just isn't any room to go in and take pictures of anything. So this is the latest photo I could find, is at 32 weeks gestation. All right, so that's it for what I have as far as creating new life. So please come on back for my last video. I'll see you shortly.